Hi, this video has a quick refresher for genetic topics related to Mendelian genetics, or in most cases, where one trait is controlled by one gene. Let's think about one of Mendel's famous experiments to review some of these topics. So, as you may remember, a lot of Mendel's work was done with peas, and he had different crosses of peas. He had a parental generation of true breeding round peas, and he crossed them by true breeding wrinkled peas. He looked at these peas and he saw that they either appeared round or wrinkled. And when he crossed these peas, he saw in what we're going to call the F1 generation that all of the resulting peas were round. They had the same appearance as the round parent. Let's review some vocabulary that we need to describe what we're going to see in this experiment. First, important vocabulary word is phenotype. The phenotype is an observable trait. In this example, the phenotypes are the appearance of the peas, either the round pea or the wrinkled pea. Next, we have genotype. This is the genetic sequence at a locus. In the locus, I'm using this word here to refer to a position in the genome. This may be a gene or it may not be a gene. Later, we're going to talk about genome organization. We're going to remind ourselves that in a lot of genomes, most of the positions in the genomes are not genes. The next important word is allele. An allele is one of two or more versions of a gene, or actually, what I should say here is two or more versions of a gene or locus. Any position at the genome can have a different genetic sequence. And in a diploid organism, that is an organism that has two copies of a gene, each one on a homologous chromosome that they inherited from either parent, we use the word homozygous to refer to the situation where the individual has two of the same allele at that locus. And we use the term heterozygous to refer to the condition where the individual has two different alleles at a locus. So let's go back to our peas and talk about this in the words that we just learned. So now we know that the phenotype and the parental generation is round and wrinkled, and the phenotype and the offspring is round. We also can start to describe the genotype. And here I am going to say that the genotype of the parents is big R, big R. I'm saying that because I know these organisms are diploid, and I know that those round parental peas are true breeding, which for us means that they are homozygous. Same for the wrinkled. Little r, I'm going to say for that genotype. I know that they're true breeding, and that means that they only have that one version of that locus that confers that wrinkled phenotype. And of course, what that is going to mean for the F1 offspring is that these individuals are heterozygous. They inherited one copy of the big R allele from the round parent and one copy of the little r allele from the wrinkled parent. The phenotype observed in the F1 generation brings us to another important vocabulary word, and that's dominance. Dominance is a relationship between alleles, where one allele expresses a phenotype that masks the phenotype of another allele. In our case, we saw that the true breeding round parent crossed with the true breeding wrinkled parent resulted in heterozygous offspring. Again, heterozygous is describing the genotype of this pea, and the phenotype of that pea is also round. And that is because the big R allele is dominant to the little r allele, or conversely, the little r allele is recessive to the big R allele. Notice that I'm talking about dominance and recessiveness as the relationship between the alleles. A lot of us have a kind of bad habit of talking about dominance with respect to the phenotype, of saying that the round pea is dominant to the wrinkled pea, And that's going to be problematic going forward in this class because, as we are going to learn, there can be more than two alleles. So, for example, there could be multiple alleles conferring that wrinkled phenotype. So, if we're always just talking about dominance and recessiveness with respect to the phenotype, we're going to get confused because multiple genotypes can confer the same phenotype. So moving forward, let's try to make sure that we are only using the words dominance and recessiveness to describe a relationship between alleles. Now let's think about what happens when you cross P's in the F1 generation. Again, the phenotype of these P's is all round, but we know that the genotype is heterozygous big R, little r. And what are we going to see when we make this cross in what we're going to now call the F2 generation? To do this, of course, we use a Punnett Square. A Punnett Square is used to predict the offspring genotypes according to Mendel's laws. So to do this, I am going to write the genotype across these different rows and columns. So we know that one of the P's being crossed is heterozygous and the other P is heterozygous as well. Those are the two genotypes of the P's that are being crossed. And now I can fill in this Punnett square. Here is the big R, reading across the column and row. Here is big R, little r, reading across the column and row. Here, we could write this as little r, big R. The order doesn't matter. It's up to you. And lastly here, we have little r, little r. Again, these are the genotypes that we expect to see resulting from this cross. To know what the phenotypes of the offspring resulting from this cross are, we have to know something about the relationship between the big R and little r allele, which allele is dominant to the other, if one is dominant to the other at all. And of course, we already know this dominance relationship from the parental cross, where in the F1 generation we saw all-round offspring. That means that big R is dominant to little r, so you're going to see that the F2 offspring with the big R, homozygous big R allele have the round phenotype, and also the F2 offspring that are heterozygous for the R locus also have the round appearance. You will only see the wrinkled appearance in the F2 offspring with the homozygous little r genotype. And because of the results of our Punnett square that we saw before, we know that there should be a 3-to-1 ratio between these phenotypes because there are 3 different ways to get around P, but there's only one way to get the wrinkled P. If you have any questions about why we're observing this ratio, come talk to me, shoot me an email, or also you might want to revisit some introduction to probability. Probability is really important in genetics. And now, I hope that some of you are wondering, why are some alleles dominant over others? How do these different alleles confer different phenotypes? And what you're seeing here is an amino acid sequence alignment of the SBE1 gene in P's. This is a starch branching enzyme that is actually responsible for whether the P is round or wrinkled. And as you can see, for at least most of this sequence alignment, we're comparing two sequences. There's the BIO336. That is what we would call the wild type, or kind of normal functioning full-length sequence of this starch branching enzyme that is going to confer the round phenotype. And it is what we have been calling big R. We have another sequence being displayed here too, and that is this, I'm going to draw over here, but it's the second one here, the JI1194. This, we can say, is a mutant allele. You might see that word used. It is not a full-length transcript. It does not make a functioning starch branch enzyme. So if you don't have that enzyme at all, then you could display the wrinkled phenotype, and this is what we have been calling little r. And what you'll notice here is that for the most part, if you just compare these sequences, and again, this is the amino acid, so this is the protein sequence of this gene, all along here, you're mostly seeing that these are really similar. So that's the first 60 amino acids. This is the next 60 amino acids. There are some differences, but it's really nothing major. And it's not until you get down here to the end where you see some major differences. And they've also, the authors of this paper, have included a few more alleles of this gene that they've observed in natural populations. And importantly, you'll see that I'm going to highlight in red here, if you follow along this wild type version of the gene, you see the complete full-length transcript. That's the complete protein. On the other hand, if you're following along with our mutant version of the gene, you'll see here that we're going along, looking the same, and then it suddenly stops. So what happens is that this mutant version of the gene is truncated. So it does not produce a full-length transcript of that for that protein, and thus that protein is non-functional. You don't get that product, and that is what results in the wrinkled phenotype. That last slide was showing us the amino acid sequence that would encode for the protein. And what you can read about in that paper is that they found that the SBE1 allele at the gene contains an insertion. So if you were not looking at the amino acids, but you were instead looking at the A's and T's and G's and C's of the genetic code, you would see that the mutant SBE1 allele for the little r allele, that gene was actually longer. It had more nucleic acids in the sequence. So one thing that I want you to think about, I'm going to stop here, is that how can a genetic insertion in the genome result in a shorter and thus non-functional protein? We'll stop for there and look forward to learning more.